All right, yeah. Welcome to our Black and Comp Bio seminar. I'm your host, Melissa Minto, and today we'll be having Bryana Harris talk to us about her research. I'll be joined by Kaylin Clark, our professional development coordinator. She'll be helping with the chat, and so if you have any questions, feel free to reach out to me or Kaylin during if you have any difficulties. Um, yeah, so today we'll be hearing from Bryana Harris. She's a rising fifth year in the Department of Biomedical Engineering at University of West Virginia of Virginia, sorry.
Yeah.
Both of both her undergraduate and graduate research focuses on cardiac regeneration, and today she'll be talking to us about her work in developing computational models to study cardiomyocyte proliferation. So without further ado, I'm going to turn it over to you. And if would you prefer people ask questions as they come up or wait till the end?
I prefer wait to the end just because I may not see them. But yeah.
Sounds good. All right. So we'll listen in, and then at the end, we'll have questions. You can type them in the chat or unmute yourself and ask.
Let me get rid of the floating. Okay. Hi guys. As mentioned before, my name is Bryana Harris. I go to University of Virginia in the Biomedical Engineering Department. And so I'll be talking about my grad research today. So systems approach to identify novel mechanisms driving cardiomyocyte proliferation. And so a little bit of background about me. So I am a chemical engineer turned biomedical engineer and systems biologist. So I grew up in Alabama, and then I got my bachelor's in chemical engineering from Auburn University in 2019. And so while I was there, so my time in undergrad, I did research in microfluidics and biomaterials. So I had two different RU experiences as well as research at my home institution, which was mentioned before. And then I started at University of Virginia to get my PhD in biomedical engineering. And so now I'm coming to the end of that road. I'm hoping to graduate summer 2024 and then starting a postdoc soon after. So a little bit of background on kind of like the basis of my research. So ischemic heart disease is the leading cause of death worldwide. And many of the patients that have this disease also suffer from myocardial infarction and also develop progressive heart failure. And so how we characterize heart failure is by the heart's inability to overcome the loss of established cardiomyocytes in an exit from the cell cycle. And so current clinical and pharmacologic therapies such as ACE inhibitors, beta blockers, LVAD, CRT do not fix the underlying cellular defects, but instead typically only prevent or slow the progression of heart failure. And so there are several strategies within the field to address this issue. One example is my undergraduate research, which is engineering cardiac tissues or cardiac tissue engineering. So a little bit of background on that and how I got started in this. So like I said before, I went to Auburn University where I was in the chemical engineering department. And while I was there, I had some family members who suffered from heart disease and heart failure, and I knew I always wanted to go into the medical field. So I sought out opportunities to do research, and that's when I found this thing called Cardiac Tissue Engineering Lab. And so kind of a brief overview of what I did in this work. So in this work, we took human-induced pluripotent stem cells. We mixed them with a biomaterial solution called PEG fibrinogen, and then we pipetted this mixture into 3 different molds. And so the main thing that we wanted to see is if what if the different geometries of these molds had an impact on the contraction of these tissues. And so we cross-linked these hydrogels with light, and then after the process, the idea was that the hydrogels would remain on the glass that we had while the mold will be removed. And so then we went into a process of differentiating these tissues into cardiomyocytes. So at about day 7, we could see initial contractions of these tissues, and then by day 21, we would see a maturing cardiac tissue with increasing contraction. And so kind of my initial kind of project of like in this lab was to take this data from different grad students that were doing these experiments and run contraction analysis using a MATLAB program. And then further on, I was able to do a lot of the experimental work. And so as I was thinking about my undergraduate work and what did I want to do in grad school, I started asking questions about the molecular basis to why these cells were growing and proliferating, and how after their differentiation into cardiomyocytes, how we could continue this process. What I soon realized was that even in these great tissue engineering studies, there is a clear deficiency in our current knowledge of cardiac biology, in that the molecular network that regulates serum renewal is mostly unknown. And so this led to, as stated by the American Heart Association, a need to identify mechanisms of indigenous cardiomyocyte renewal in mammals that can be targets for therapy, which includes mechanisms of cardiomyocyte proliferation. And so with these things in mind, I looked into ways that I could do work to combat these issues. And so this led me to cardiac systems biology, where we have 2 main objectives that we like to think about. So the basic science objective pertaining to my work is to identify molecular networks and control points to decide how the heart will proliferate. And then the more translational objective is to rewire these molecular networks to prevent or reverse heart failure. So this is taking computational models and then performing quantitative experiments, and then just repeating the process to create a more robust model of cardiomyocyte proliferation. So this leads me to more into my graduate research, which is engineering molecular networks for cardiomyocyte proliferation. And so the main goal here is regeneration, and how we want to do this is through proliferation. So we have a cell, we want to encourage the cell to re-enter the cell cycle and divide to make even more cardiomyocytes. So there have been several candidate regulators found over the years to regulate cardiomyocyte proliferation. And these are things like non-coding RNAs, such as microRNAs, cytokines like neuregulin, FGF, signaling transduction pathways, like the Hippo pathway that is very important in the well-studied pathway within cardiac and even cancer biology, and then cell cycle regulators like E2Fs and cyclins, those things that are increasingly driving DNA replication. And so, although many candidate genes have been discovered to stimulate CM proliferation, these studies alone do not give us a great framework to identify and prioritize one stimulus over the other. And so, like any other complex biological system that we run into, there is a need for innovative computational models and quantitative analysis to understand the interconnections between various regulators of CM proliferation. And so this brings me to my overall objective, which is to use in vitro proliferation assays and systems biology approaches to identify potential therapeutic mechanisms that regulate cardiomyocyte proliferation. In this way, we were basically taking all of the important factors that we can find within literature pertaining to cardiomyocyte proliferation and bringing them into one process where we can look and see how these things are interconnected. So to start, I built a model of the cardiomyocyte proliferation signaling network where I use approximately 30 literature articles describing connections between various regulators and their effects on important proliferation processes like denaturfication, mitosis, and cytokinesis. And then we also use secondary public resources such as KEGG, SIGNOR, and cell signaling. So our goal was to kind of look through cardiomyocyte literature and see if there were any connections from literature that we could use to build this model. And so building the model came from cardiomyocyte literature, but we can also expand into other cell types just to make that connection. And so overall, this model incorporates 6 different modules that detail important pathways regulating CM proliferation. And so we started with the growth factor module, and these are things that stimulate cell growth, survival, and differentiation in tissue repair. So things like neuregulin, FGF, IGF, and their process in regulating proliferation. We then moved on to the HIPPO module, and this describes this conserved pathway that is known to control organ size by regulating proliferation and cell renewal. And kind of what I mentioned before, and I'll probably mention again, just because it is important, this has been studied a lot within cardiac and even cancer literature. And then we moved into describing the importance of events of proliferation, starting with our denaturfication module. And this describes a lot of the regulation of different cyclins and cyclin-dependent kinases and their complexes, and even cyclin inhibitors and how they affect denaturfication. And then we did the same thing with the mitosis module, where we found important components driving mitosis. One important one is cyclin B, CDK1, as well as some similar things from the other modules. And then we did similar things with the cytokinesis module. And so what's important to note here is that the information is very limited on cytokinesis and how it's affecting CM proliferation and how that process actually works. So we kind of had to expand out and kind of dig deeper to find these particular connections and how this is actually working. And then finally, we created an output module because we know the important outputs of these different modules, but we also wanted to show that how they're dependent on each other. So mitosis needs the effects of denaturfication in order to complete, and then so on and so forth, so for cytokinesis. So we wanted to really show the dependency of all these phenotypic outputs on one another for the process of proliferation. So after we built the model, we wanted to convert these relationships that we had into what we call logic-based differential equations. And so this approach is for cell signaling networks, is based on normalized Hill activation and inhibition functions controlled by logical and OR gate operators to characterize signaling crosstalk. And so this allows for quantitative predictions of network functional relationships and dynamics. So the dynamic activation of each node is dependent on the logic of its activating or inhibiting interactions and on the time constant that we have, which is a default parameter value based on the type of interaction that we are seeing. And so there are various different tunable parameters and default values that we use through this modeling method that we can change and adjust based on what we want to look at and what experimental basis we want to look at. And so I do want to mention that this is a public available resource through GitHub. It's called NetFlux. And if anyone has any questions, I can discuss more at the end. So one of the great things about these models that we build, and specifically this one, is that we can create and predict potential experiments and then validate them with actual experimental data. So an example we have here, we simulated the effect and increased input of neuregulin in combination with p38 inhibition would have on denaturfication. And so what we can see that compared to our control state, there is an increase in both cases. So in both cases of neuregulin by itself and then with p38 inhibition, and the same can be seen for FGF1. And so to validate this prediction, we looked in cardiomyocyte proliferation literature and pulled experimental data from Engel et al., where they treated neonatal cardiomyocytes with neuregulin and FGF1 in combination with p38 inhibition and observed the effect it had on BRDU positive neonatal cardiomyocytes. And this is basically saying the same thing as like percent denerification cardiomyocytes. And as you can see from this data, it shows similar trends to what we are seeing from the model predictions. So this is just one example of kind of our validation process for the model. And so what we want to do is we want to continue this validation process. And then we did that by first focusing on important phenotypic outputs. So denerification, mitosis, cytokinesis, and we even added in polypoid binucleation cells. And then we moved on into specific regulators being studied in cardiomyocytes. So things like the different cyclins and then the cyclin inhibitors. And so overall, our model predicts 73 of the 78 experiments from literature, giving us a 93.6% validation accuracy. And so it's important to note when we were validating our models, we stuck specifically to cardiomyocyte proliferation literature. So all of the experiments done within cardiomyocytes. And at the moment of finding the different validations, we weren't as concerned about what cardiomyocyte cell type, but it is important to note that almost 85% of the validations that we did find were in neonatal rat cardiomyocytes. And that's just how cardiac literature is going in most of the experiments that are done. So next, we wanted to do a virtual knockdown screen to predict regulators of cardiomyocyte proliferation. And so how we do this is we take individual nodes of the model, we knock it down, and then observe the effect that knockdown has on all other nodes of the model. So this is basically taking one of our default parameters, Y max, which is defaultly set at 1, and then we're knocking it down or decreasing that value to around 0.1 or 0 to simulate a knockdown of that particular node. And so in brief, each column is the response of all proteins and outputs to the knockdown of that protein. And then each row is the response of that protein to the knockdown of each protein in the network. So we have influence of the node and sensitivity of the node. And so overall, this is just a shortened version of sensitivity matrix because if you remember, my model has 73 different nodes. So overall, this gives us roughly 5,000 different measurements. So 5,000 different potential experimental measurements that we could run. So to dive more into what we can see from this virtual screen, we can look to cytokinesis. It is one of the most important parts of the model and the overall process of proliferation, but it's also one of the least studied with the connections, kind of as I mentioned before. So if we look at the network with a total knockout screen for cytokinesis, and this basically means that we are knocking down each node of the model one by one and observing the effect it has on cytokinesis. So if we do this with the basal state or the basal form of the model, we have a few nodes that pop up, things like PKA, LOX1, RB1, SPA3, and GSK3 beta that show up as important or influential nodes. And in fact, when these nodes are knocked down, they are causing an increase in cytokinesis activity. So if we look at how cytokinesis will respond in the context of increasing the input of two different regulators, neuregulin and YAP, which are important and studied regulators in cardiac literature, we can see that in high neuregulin state, there are nodes that have positive and negative effects on cytokinesis, and these are briefly things like cyclin and the cyclin inhibitors, and things like APC and P38, things like that. And then if we look at a few particular nodes, we can see how these nodes change influence in the presence of different signaling contacts. So we can see how P53 goes from being very important in increasing the factor cytokinesis in high neuroregulin state, while in YAP, it's not as important or having as big of an effect there. So continuing with this idea that signaling context is important, we wanted to look at the global influence in the signaling context. So starting with the basal state as our basis, we wanted to see how the influence changes in these different contexts. So in the neuroregulin context, we can see that increase of the growth factor influence, which is expected to change because neuroregulin is part of that module. And then we have a corresponding increase in the DNA replication, mitosis, and a very small increase in the cytokinesis module. But what is really interesting is that when we increase neuroregulin or have a high neuroregulin state, we have the hippo module kind of decreasing in its influence. And then we look at the high YAPS context, where we can see an increase in the hippo module, which is expected, with a corresponding increase in growth factor, DNA replication, mitosis, and even the cytokinesis modules. And this is kind of showing us and insinuating that there is some type of crosstalk that is happening between the hippo and growth factor modules, and that's kind of represented here with these different arrows. So next we wanted to see if we could, and as I've reiterated so many times before, is important in regulating proliferation and has been studied a lot extensively in cardiac literature. But what is still unclear is how a YAP is exactly working downstream in cardiomyocytes. So what we did is we did a simulation where we took some important nodes, looked at their activation in the basal state of the model, and then we simulated the effects of a drug known to increase proliferation through the YAP-TEAD complex in cardiomyocytes. And so what we found was that there was a significant increase of c-Myc FOXN1 PI3 kinase, as well as a significant increase in our phenotypic output, so DNA replication, mitosis, polyploid. So using the results from the virtual screen, we decided to kind of hone in even more into the hippo signaling pathway, specifically how YAP is inducing proliferation. And so out of all the components, like I showed before, that were influenced by YAP, we found that FOXN1, c-Myc, and PI3 kinase are the most important. They are not only targets of YAP itself, but also have connections to mitosis and DNA replication modules. So we took this information and created an experiment with our model. So we asked, what if we treated with a drug, the TT10, known to increase proliferation through YAP-TEAD, and then measure how these three hubs, if inhibited, would affect that base activation. So what we did is we have a control. So just like the base activation DNA replication activity in the model, and then we simulated the treatment of TT10. So just increasing the input weight of that, where we got a base activation of about 70%. And then we went through each hub, and then we said, okay, if we inhibit the hub alone, but then we inhibit it in combination with TT10 treatment, how does that affect that base activation of TT10? And what we can see, there is a decrease in all three hubs when we are inhibiting these three different hubs. And the same can be seen in mitosis activity. So the next thing we wanted to do was to take this prediction and then do experimentally validate this prediction of PI3 kinase, c-Myc, and FOXN1 mediating YAP signaling. And so what we did with this, so how we kind of work through this process is we isolate primary cardiomyocytes. We plate them in 96 flow plates. We exchange media for a couple of days, and then around the third day, we treat the cells with our conditions, with our drugs. So in this case, it was TT10, so our positive control drug with the three inhibitors, PI3 kinase, c-Myc, and FOXN1. And then 48 hours later, what we wanted to do was fix, stain, and do high content imaging. And so what we did from those images that we took, we did automated image analysis, which then gave us two important outputs. So the percent Cas7 nuclei cardiomyocytes, which is representing our DNA replication, and then percent phosphohistone 3 positive nuclei cardiomyocytes, that is representing our mitosis. And so from there, we can see images from that experiment that I did. So this is just representative images. So we have DMSO and TT10, our negative and positive controls, where we have DAPI, which is our blue nuclei stain. Then we have Cas7 or phosphohistone 3, which is our magenta that you can see here. That's very visible. And then we have cardiac troponin T, which is just our cardiac stain. And so what we can get from all of that data, so we did the same thing you saw with the model predictions, right? So we have kind of like the basal states, the normal state of positive Cas7 cells and phosphohistone 3 cells, so around like 10-20%. And then we treated with our TT10, which gave us around 68% base activation. And then we did the same thing we did with our predictions, where we did the inhibitor alone and then in combination with the treatment of TT10 for all three different hubs. And what we can see, there's a significant decrease with the PI3 kinase and c-Myc inhibition. The same can be seen for cells positive for phosphohistone 3. So that's really cool. So it's predicting, so we're validating what we've predicted from our model. And so we wanted to take this a step further. So we wanted to see if there was any in vivo data that can confirm what we're seeing. So what we did is we searched through literature, and what we found from Monroe et al. Is where they overexpress YAP5SA transgene in adult cardiomyocytes. And then they did two different measurements. So they did ATAC-seq and RNA-seq. And so if we look to the right, for the RNA-seq, we can see that MYC and FOXN1 have significant differences between control and then the YAP5SA overexpression in their relative mRNA abundance. And then we look at the ATAC-seq data, where we can see that the YAP5SA overexpression induces an altered open chromatin state within MYC and our PI3 kinase that we have here. And so what's important to note is the one that's consistent in both measurements is MYC. And so that leads us into potentially like working with collaborators or getting more information on how MYC is actually working with YAP to stimulate cardiomyocyte proliferation. And so in summary, from this project, we developed a predictive model of cardiomyocyte proliferation signaling network that can identify crosstalk that regulates the proliferative response of cardiomyocytes. We predicted crosstalk from the Hippo module to the growth factor module involving three network hubs, so PI3 kinase, c-Myc, and FOXN1, and their mediating effects of YAP on cardiomyocyte proliferation. And then we were able to validate these experimentally. And then on top of that, we were able to pull in vivo data that showed that YAP5SA overexpression induces increased not only mRNA expression, but altered open chromatin of MYC. And so if you have, I'm free to answer questions after this, but I also have a preprint out and that you can find and then get more information on this work. And so kind of just a few contributions to this particular field. So this is creating the first large-scale computational model of the signaling network regulating cardiomyocyte proliferation. And this is using a logic-based ODE method, which is integrating dozens of different studies across laboratories into one network structure. And this will provide us like novel perspectives in signaling crosstalk that regulate cardiomyocyte proliferation dynamics, where we can incorporate more data as it comes along and work with collaborators when they find interesting things. We can apply it to this model and then make predictions and even validate those predictions. For instance, we have a collaborator that found something interesting where I took those targets, applied it to my model, and now we're working to make predictions where they can validate what is shown by my model and get some interesting stuff from that. Just briefly, some future directions in my grad research. You saw this earlier when I first began talking about this project. So kind of like the next steps and kind of what I'm working on is kind of understanding the cell cycle process and using single cell imaging data to kind of predict what is happening in death with the cardiac cell cycle. And then also just identifying more potential modifiers of these network modules that regulate proliferation and integrating them into this model and kind of building a more complex and robust model for cardiomyocyte proliferation. And then some like of my future directions. So as I mentioned before, I'm hoping to graduate in summer 2024 and kind of thinking about what I want in postdoc relevant to kind of what I would want in a future lab. So I'm hoping to incorporate a lot of the system biology approaches that I've learned so far and a lot of the computational methods that I've learned and incorporating those into more in vitro experiments. So more experimental experience, and then applications to human disease. So working with different patient samples and clinical perspectives, and then hopefully even applying some health disparities stuff to my work eventually. Oops. So I know I ended kind of early. I didn't want to make it like too dense, but anyway, so I like to thank my lab, my PI, Jess Saucerman, and our collaborators, and then NSF and NIH. And then I have my email here, free to answer questions now, but if you have any other discussion items, there's my email there. Thank you.
Oh, thank you so much for that wonderful presentation and such incredible work. You really that was a lot of work that you just discussed. So, yeah. If anyone has questions, please feel free to hop off mute and ask now.
I do have a question. First, yes, great presentation. It shows a lot of different pieces that are involved in here. And I think one of the questions you actually answered, because it's so many networks that you're looking at right now, but that's one of the goals, to have like this large network first to kind of get a landscape of the land. And so I'm wondering, in all the studies that were incorporated, the models, were any of them human studies, or were they all rat or mice or other animal studies?
They were majority animal studies, so there are a few studies that were kind of human iPSC derived cardiomyocytes, but there were like very few that we could use for this. But yeah, I would say about maybe 3 to 5% for that. But yeah.
Okay, and then because I'm wondering how do we then extrapolate it to human populations? It's something that I'm working with as well. A lot of like rat studies looking at fertility, and it's like, how do we get to, because we show that maybe we can replicate what we're seeing experimentally, and then and linking it to the human health effects or the health outcomes. Is there any specific pathway that you're particularly interested in, or that could help make that leap to a human health effect? Or that even maybe it's for your future directions, is there something that you're mostly interested in?
So that's a kind of like a quizzical thing where it's like you kind of want to apply it to the human basis because that's like the goal, right? We want it'll be great to have like a human, like validated computational model of this stuff. But then it would just take like extensive work by either us or different collaborators. So as far as another pathway, so not a pathway in particular, but I know my lab, and they'll probably get onto me if this is wrong, but I know we're starting to look into kind of more of like differentiation and kind of what's driving that, especially in terms of using more HIPSC derived cardiomyocytes and kind of like the pathways that are important for that aspect. And so if anything, like future wise, I would think kind of looking at strictly the pathways that are driving that differentiation process, which I think includes some of like the different growth factors, and even things from like the hippo module or pathway. So what's driving that differentiation, and then kind of focusing in on that, and then hopefully once we understand that, then making it a more human-based model, if that makes sense. Because I know there's like human cardiac cells out there that we could buy and then do, but then there's also issues with that. But yeah.
Okay. Thank you so much.
Yeah.
And maybe it'll come up as you're asking. I am interested in Netflix, but
Okay.
if you can
Yeah.
go back to that at some point. Yeah. But thank you.
Yeah. Yeah.
We also have a question in the chat from Elise. She says, fantastic and such impressive work. She's wondering if you saw any sex differences in mRNA or chromatin states for MYC or PI3K, and if you typically see or look for sex differences when modeling molecular mechanisms of disease.
Yes. So no, we unfortunately within like the open chromatin data that we pulled, we weren't exactly looking for sex differences, but that would be an interesting thing to ask those who like did the in-depth like data for that project there. And no, we don't specifically focus on sex differences. There have been a couple people in my lab interested in doing so, and trying to do work more on like the fibroblast side and looking at sex differences. But I can see where it would be like a possibility to do that. But I also think there's like a lot of things like before that we need to understand more about cardiomyocytes before we can like really focus in on the sex differences. But I think that would be really cool. And I actually just went to a conference where sex differences within the heart is a very big deal, and more people are doing studies on that. But no, I haven't applied or really actually read anything focusing on like sex differences in cardiomyocyte proliferation exactly.
Okay, and we have another question from Elfrida Silva. Hopefully I'm pronouncing your name right. And they ask, in which proportion is this model accurate, knowing that some analyses are based on animal cells?
Sorry, I had to go find the question. So accurate as in, so accurate for cardiomyocyte proliferation in terms of neonatal, like rat cardiomyocytes, yes. And so it would take kind of like what we kind of were discussing beforehand. It would take a lot of like extensive studies in humans and using human cells, and then even like revalidating this model almost completely to get a more human-based cardiomyocyte cardiac model. And so to say whether it's accurate or not it's kind of like hard to say, but we just kind of keep to the regulators that are conserved across species. So I know a lot of work has been done to look at compare like HIPSCs to neonatal rat and adult rat like cardiomyocytes. So I think like a lot of like RNA-seq data has been done towards that, just kind of showing like which pathways are conserved across that, and then applying those pathways to the model, and then doing, like I said, more in-depth like human cell work for that. I hope that answers that question. It's kind of like a hard question to like answer in that it's just the state of kind of the literature that we have for that. So
yeah, I think it's awesome that you put together. Well, I'm just out of curiosity, how long did it take to like come up with the network, and like what was your inclusion criteria for including this in the network versus not?
yeah, so we kind of knew kind of like the important like aspects. So growth factor, hippo, like those are kind of like already laid out for us in literature. So then it just became about like, okay, what are the connections? Put like whatever the connections are, let's put it down on paper. Let's put this like in our model. Let's make all these things. And then kind of going back like piece by piece and saying, okay, we found this connection, but is this connection a consistent connection within cardiomyocytes or even other cell types? Are people saying like AKT is connected to PI3 kinase? Like that's pretty well known and pretty conserved across literature. So like just looking and seeing like, are these things consistent? And then if it's not, kind of rethinking whether it's important to have it there or remove it. So it's kind of like how much information can we find about that connection? That makes sense.
Yeah, that must have been a lot of hard work, just like coming up with the network to begin with. We have another question from Elise. She asks, if you have any quick advice for someone that would like to learn about modeling and or ATAC-seq data.
So I'll start the ATAC-seq data. So I also don't know much about it. I thought it was just cool that they did this measurement, and then we were able to get the assistance of a collaborator to kind of help us analyze that data. So as far as like learning more about ATAC-seq, I would just look for labs that are doing that work and just kind of like reaching out and learning more information from them. And then I know there are a lot of resources online that gives you kind of like a base understanding, and that's kind of what I did. As far as modeling, so one weird thing, so when I was an undergrad, as a chemie, we did have some courses where we had to use MATLAB and computer stuff. And so I had one teacher who did modeling at my university. He taught one of our classes that was almost fully like computer stuff. And I took this class and I even told him, I was like, I'm never touching MATLAB again. I'm not doing this anymore. I will never do what you're doing. Guess where I ended up? I ended up in a computational modeling lab. And then from there, I kind of like learned from this lab and my PI how to do it. So I would just suggest like just reaching out. If you're interested, there's tutorials online. Like I said, Netflix is like an open source. And there's a lot of different faculty members who love this, like my PI, and who loves to teach it. So yeah, just look for those opportunities. Yeah.
Awesome. Maybe that's something we can try and do in the future, have a Netflix demo.
Yeah, that'd be
So I'd
great.
be
Yeah,
interested in learning too.
we do. I know we had like a quick week seminar last year, so I think we're doing that again this year. So I'd be happy to share that information. But
Yeah, that would be great.
yeah.
I had a question about the genes, like the YAP and the neuroglin that were coming up. Do you find that like in human population that there are SNPs or mutations in those genes that were coming up as like highly influencing your models?
Can you repeat that question? Sorry.
Sure, yeah. So for the genes that had high influence in your models or your modules, did you find that those genes also had SNPs and mutations, or mutations for like people who had cardio-related diseases?
So you're just asking like whether these like important nodes were also important in like different like disease states.
Right, yes.
No, so I didn't look specifically at that, but that would be interesting to kind of like decipher and to know as to like whether when like different labs are doing like these disease models or like animal studies where they're causing this disease or causing this infarction, kind of what's popping up is most important and kind of applying that to the model. And so like the model that I created is a very general one, but it would be great to make it a more like, I guess like the disease relevant like model. So if we had a particular heart disease that was very important and then applying that to the model, that would be really cool to see the effects. And that is something that we could do with the data if we had it. But yeah, no, I didn't specifically look for that. So no.
Yeah, that would be a cool, like, that could be the next paper, you know?
Yeah, yeah, that could be a whole another paper.
Yeah. Are there any more questions in the audience? All right, well, I just want to say thanks again for taking the time to share your work. It's beautiful. I'm going to go through and read the paper again because I really, I've never done anything like that. So it was really nice to hear from you and hear how it was done. And so thank you.
Yeah, of course. And if anyone has any more questions, feel free to email me and then get in contact with me. So yeah.
Yeah, and before we close out, if anyone in the audience or people in the audience could fill out our feedback poll for this event, just go on and let us know how you like things, what we can do better, what you'd like to see, like a Netflix demo, and yeah, so we can continue to give the community what they want to see. And then we also have a new podcast out with Dr. Javon Carter. He's talking about his experience in computational biology in a nonprofit research institution and overcoming imposter syndrome. So go to wherever you listen to podcasts and check that out. All right. Thanks, guys. Bye.